So I will start off. I was going to go through and introduce everyone to make the meeting flow better and keep things going. So I'll do all the introductions here. First off, we do have Dr. Mark Reiter. Excited to have him join us today. He is from Virginia Tech. He is the Associate Professor of Soils and Nutrient Management. And he's also the Director of the Eastern Shore Ag Research and Extension Center. So happy to have him here. He's going to be discussing optimizing potato nutrient management and soil fertility considerations. I know this is a very important topic. We've been looking for somebody to fill this need and talk about this, and I know Mark will do a great job. Excited to have him with us. After that, we have Dr. Craig Yencho. He is the William Neal Reynolds Distinguished Professor and Program Leader of Sweet Potato and Potato Breeding and Genetics Program. And joining him will be Mark Clow, who is the Research Scholar and Extension Associate who runs the Potato Breeding and Genetics Program at the Vernon James Center in Plymouth. They're going to be talking about a potato breeding program update. After that, we're going to hop into talking about disease management. Thankful to have Dr. Jean Restaino here with us. She is also a William Neal Reynolds Distinguished Professor of Plant Pathology, and she is the Director of Emerging Plant Diseases and Global Food Security Cluster. So going to be talking about disease management. And then also extremely excited to have Dr. Anders Huseth, who is the Assistant Professor and Extension Specialist in Department of Entomology and Plant Pathology. He's going to be talking about insect management. So I know a number of these people, if you've been to our meetings in person over the last several years, you've got to know them and hear them speak, and happy to have Dr. Reiter join us as well. And with that, I will turn it over to you, Mark, if you want to go ahead and jump right in.
Thank you, Austin. Thank you for having me today. So I'm sorry I'm not there in person, of course. We much rather have these meetings in person, but we're so happy to be able to participate via Zoom with you. So what I wanted to do today was just to talk a little bit about some of the soil fertility considerations that we have in Virginia, and more specifically for the Eastern Shore of Virginia, where I live and work. And a lot of our focus over the past few years has been dealing with some nutrients that are, of course, of concern to the Chesapeake Bay, as far as pollution, and really honing in on extension recommendations for improving productivity, while also making sure we're not over fertilizing, of course, for water quality standards. And then some considerations dealing with working with Steve Rideout and plant pathology with just better tuber quality and reducing rots in our production systems. So one of the slides I always enjoy showing, and probably my favorite slide I've ever made, was dealing with Liebig's Law of the Minimum. So every time we think about soil fertility, we want to make sure we understand that we're only putting out the nutrients that we need when we need them. So we're really filling in these gaps that may decrease production or quality, and also to make sure we're getting the most bang for our buck. Now, I know certainly with potatoes that fertilizer is a pretty small part of the overall production budget, but still it's an important thing to consider, and could be one place you can shave off a few costs if you don't need that fertilizer. Or certainly by adding some of these nutrients we're going to talk about, you might reduce rots or potentially improve your tuber quality. So what this Liebig Law of Minimum means is if you have every nutrient in your soil, if you pretend it's a barrel of water, let's say in this case, the lowest slat or the lowest nutrient that causes limiting production, of course, will reduce crop production and reduce yields. So in the case demonstrated here, nitrogen is the limiting nutrient. So what we would say is, okay, per Liebig's Law of the Minimum, if nitrogen's our limiting nutrient, you patch this hole, then what's our most limiting nutrient? Well, then we start focusing on phosphorus. Patch that hole. In this example, calcium is the most limiting, until we get to the point where the nutrients are no longer limiting, and frankly, it's somebody else's thing to think about, somebody else's problem, as I would say at our station. So I know mostly in our production system, certainly in the mid-Atlantic region, overall, water is our most limiting element that we worry about, but certainly nutrients, especially as nitrogen, can be problematic as well. So this is what we're going to focus on, is finding these nutrients and thinking about some of these nutrients that are not commonly issues in crop production systems historically. And one of those nutrients, just to give you a hint from this older graphic that I stole from the textbook, is sulfur. So if you think about some of the things that people used to think about from this older example, everybody's always focused on nitrogen, then phosphorus, potassium, and so forth. The sulfur was never really a consideration or an issue. So we'll just address some of that as well as they relate to potato production. So when you think about potato production and fertilizer, I know we always have big fertilizer bills, right? Fertilizer is really important. And so if you think about why is if you're shooting for some top yields, 300, 350, 100 weights and so forth, excuse me, you need a lot of fertilizer to make that happen. Just physiologically, you need those nutrients to be available to produce amino acids and proteins and tubers and so forth. And so looking at this chart, for potatoes at 350 100-weight yields, you need 188 pounds of nitrogen to be taken up in that plant, and 112 of that is removed in those tubers themselves. 70 pounds of phosphates taken up in that plant, and 53 pounds are removed in those tubers. And I want you to keep this in mind, this number, because that'll be important here as we talk about phosphorus. 280 pounds of potash or K2O are taken up, and 196 pounds are removed in those tubers. So just looking at this, we can understand why every single year we're needing to go behind potatoes, or before you apply your potato fertilizer, really think about this nutrient management program to make sure we're supplying this a pretty large amount of nutrients for these top yields. And here's all the different crops. You can go back in the recording and take a look and see how that whole crop production cycle and system may change depending on what crops you use in rotation. So what I wanted to focus on today specifically regarding these nutrients are some of the main things that we think about with potatoes. Now, nitrogen is one thing that we talk about a lot, but of course we have a good handle on that. That's something well established. And we have extension publications and farmers are doing nice nitrogen management practices already. And here's a nice example of what happens if you don't have enough nitrogen fertility. This is a check row within one of my research trials. And certainly nitrogen is needed, but because of that, it's a pretty easy thing to see that's well understood. So we're not going to talk about nitrogen. And potassium as well, we're pretty much just following soil test recommendations. And it's also not a polluter. It's not an environmental nutrient of concern. So we're not really going to focus on that. But what we are going to talk about is water quality and or phosphorus and how it impacts water quality, and why phosphorus fertilizer might still be important even if your soil is testing very high as far as starter fertilizers. We'll mention a little bit about sulfur, since the Clean Air Act has basically reduced all the free sulfur that we get as far as fertilizer, and also about applying gypsum or calcium sulfate as a soil amendment. And then finally, a little bit about at planting versus banded. So over the past few years, a lot of farmers have really tried to get away from at banding or in the row fertilizer applications because it slows down planting, it corrodes your planters and so forth. And so one of the main questions we had on the shore was, could we really get rid of these in-row applications of fertilizer and just use broadcast applications? So I'll present some of that data today as well to show what we found out about that. So first, speaking about phosphorus, there's been a lot of work done on the shore as we've worked with, cleaning the Chesapeake Bay, the watershed implementation plans and so forth. And this has received, again, renewed interest as we're trying to meet our EPA TMDL, or total maximum daily load phosphorus guidelines for the Bay. And there's a lot of questions pertaining to phosphorus as far as, how much do we need, which I feel like we've done a good job in our soil test guidelines, but one of the more important things we're trying to understand still and continuously, which has been worked on for the last 10 years or so, or gosh, probably 15 years or so on the shore, is why are potatoes still responding to phosphorus positively with higher yields when we already have very high soil test phosphorus concentrations? And this is pretty important to understand, as we are still recommending phosphorus starter applications, even though our soil test for pretty much every other crop in Virginia says, no, you don't need it. But these vegetable crops still need starter fertilizer to keep us from cutting down yields, reaching our yield goals. So this is some work that was done, again, across the Mid-Atlantic, and it's been consolidated into our Mid-Atlantic commercial vegetable production recommendations, which I know y'all have one you use with North Carolina. There's a Southeast version, and where Virginia's Tech's part of that one as well. But we have the Mid-Atlantic one as well, that we really have honed in some of these specific guidelines that relate to Virginia production. And this is free and available online if you just want to go to your favorite search engine and search the title here. You can find a free PDF copy of this guide if you want to take a look at what we're doing across the board here in Virginia. So just a few things to note as far as fertilizer, and which to show where our data has been used from these studies is, like I said, nitrogen management's pretty well understood. We have a recommendation, for total nutrients of 150 to 180 pounds per acre of nitrogen for what we call a typical system. But if you're high yielding systems, greater than 250 hundredweight per acre, we have a different recommendation, which we have a publication that explains that. But related to this talk today in phosphorus, I wanted to show you how typically, if you're testing very high for soil test phosphorus, most states are recommending no phosphorus applications because that is the traditional standard soil test guideline. Because if you have enough phosphorus in your soil, we are assuming that that plant can take it and meet maximum yields. However, through some of our work, we have found that this is not true for some of these vegetable crops like potato and say tomato is the other main crop, where we still recommend phosphorus fertilizer recommendations even if we're testing very high. And so in our guide, we have corrected this by saying we really don't need any, but in Virginia, per our nutrient management plans, we're still recommending 50 pounds per acre of P2O5 or phosphate fertilizer for maximum yields. And one of the ways we're justifying this application, if you remember, if you have a high yielding system, we're removing about 50 pounds of phosphate fertilizer with our tubers anyway. So basically we're saying we're not increasing soil test phosphorus. We're not going to increase pollution potential. We're basically allowing a crop removal rate so our yields are not reduced from this kind of unique situation that we're seeing with potato crops. So this work was done over several years, and it was just a kind of a study up and down the eastern shore of Virginia with many different site years, looking at different phosphorus rates, pretty much a simple phosphorus rate study comparing banded applications or broadcast applications to banded applications of fertilizer under the row. And generally, it's a lot of data to digest, but what I wanted to show you here is there are several cases where the Mehlich 1 soil test phosphorus, or your soil test phosphorus in parts per million, was 242 ppm, which is really high, nearly 4 times what we recommend is our very high cutoff for Virginia. We still saw a yield increase in potatoes. Some sites, 120, we saw some cases where we had yield increases. And especially one location, which had 120 ppm phosphorus, all 3 rates saw yield increases. So certainly there's cases where we're seeing yield bumps and benefits from these phosphorus fertilizer applications, even though soil test said it wasn't really needed. And so when you go through all the gyrations and averages and so forth, kind of summarizing all of that data from that previous graph, when you look at yield and hundredweight per acre over phosphorus rates, we found that, all across all sites, all averaged together, at least 25 pounds of phosphorus fertilizer per acre would give you a nice yield bump that allowed us to say, hey, if we don't recommend phosphorus, if we have very high soil test phosphorus concentrations, we're going to effectively decrease yields. And there's a lot of different reasons for this. We're still working on this. Actually, I have a graduate student we just hired to focus on these phosphorus dynamic soil interaction concepts. But largely you think about how small a potato root is in comparison to some of these other crops. Potato roots, tomato roots are typically 90% of the roots are stay in the top foot of the soil, where corn, soybeans, wheat, they can scavenge unrestricted 3, 4 feet deep or so in the soil. So we think that's the main reason for this. But regardless, there's a lot of need for phosphorus as a starter application in these systems. Another study done on Delmarva, this was done by Gordon Johnson at the University of Delaware. He looked at grower fields, just kind of doing a survey saying, what happens if you do have different soil test phosphorus concentrations? Is it changing anything? And the long answer is yes. So some other data we've used to work with these phosphorus needs and kind of validation on how we can recommend phosphorus because it's been removed from the system is if you look at soil test phosphorus in parts per million on our x-axis here over our tissue concentration, whether it's leaf tissue, which is this blue line, or tuber concentrations of phosphorus, which is this green line, we see that as soil test phosphorus increases, the tuber phosphorus concentration is also increasing. And so if you think about adding the starter fertilizer to, say, a 200 ppm soil, we're removing actually more in that tuber than we would at, say, 100 ppm soil, which is good from the environmental standpoint as far as moving more phosphorus off that land, but it also still allows us to achieve maximum yields overall in these crop production systems. So moving on here to the next study where the Virginia farmers have been really asking, can we get away from putting out fertilizer with our planter? That's just too slow. It's corrosive. It's just causing some issues. I'd rather spin it on 2 weeks ahead of time. Especially the past few years as we've been wet and planting has been an issue. And so we implemented this study where we looked at as a simple application method study where we compared under the row to a broadcast fertilizer application across our plots. And then we went ahead and did do this at 3 different rates at 30, 60, and 90 pounds of phosphate per acre. And one thing to note here is this was at a 108 part per million plot averaged over the years, which were very high. So we always did this on a very high soil test phosphorus concentration. But so overall question was, did it matter? As far as these nutrients. Now, one thing that was a little different in this study is we wanted to use 10-10-10 just because we wanted to also see about N and K. So it was applied on a phosphorus rate, but with that, the yields were sometimes a little lower because nitrogen was also limiting as well. But regardless, the take-home message here is looking at this under the row versus broadcast across our plots. It doesn't matter. Visually, we didn't see a lot of difference. The plants were all healthy, lapped the row quickly. They looked really nice, whether they were banded in the row versus broadcast. But when you start looking at the data, again, this is averaged across all those NP and K rates. And so the yields are a little bit lower. But with that being said, across all those rate averages, we have our 2 methods here. Broadcast, which is, in our case, we've spread them on the day before our plots, before we plant it, versus banded, which was, again, put under the row. Generally, we found no differences. So when we've been doing this the past several years, in 2016, statistically, we did see a great fertilizer benefit, of course. No fertilizer was 9,300 pounds per acre. But whether we banded it was 238 hundredweight and 240 if we broadcast, and they're the same. So it didn't matter if we banded or broadcast. In 2017, didn't matter if we broadcast. 2018, didn't matter if we broadcast or not. So 2019, we did see a statistical response, but with that being said, this was a bad year. Potatoes were planted late. Because it was so wet, we ended up harvesting late. And I'll show you the rot slides. We had a lot of rot before we could get in there and get these plots out. So long story short, I put this in here for another reason. But statistically speaking, if you are in a bad year, Bandit had a slight advantage. So I wouldn't certainly focus on that at all because, we're talking 108 bags versus 94, not, worth talking about. But in decent growing, good, normal years, there was no benefit to putting out fertilizer with the planter versus broadcast. So we were pretty happy to see that as far as allowing our farmers to more quickly apply fertilizer over the growing season. Looking at rots, again, it really didn't matter. We harvested rots the best we could with our, just sorting nice potatoes from rot potatoes in our plots. No difference except in 2019. there was no difference, but do note that we did have a lot of rot that year. And I know we lost a lot in the field just because we couldn't get out there. So, no benefits really from rot percentages. And overall, really no benefit or differences from tuber sizes, looking at broadcast versus banded. So, looking at all this data, generally speaking, as long as you're putting out the right amount of nutrients, it doesn't matter if it's put down with the planter or not. In these cases, over these at least 3 nice growing seasons we've had here on the eastern shore of Virginia. So the last thing I wanted to talk about here real quick was sulfur. We've had a lot of questions about pretty much every crop in Virginia. We're doing a lot of work with soybeans right now with sulfur. And the potato growers had the same question, thinking about the way potatoes grow. Since we used to have around 20 pounds of sulfur raining on us for free per year, and now we basically don't. So we had about 20 pounds here via acid rain in 2000. Now we have basically none due to the Clean Air Act. And we know that potatoes need a significant amount of sulfur for crop productivity, just like any other crop. It's part of amino acids, which you need to make proteins, which you need to make your potato tubers. So, and we also know that sulfur leaches. So this was a soil pit we did at the Virginia Ag Expo in 2016, where it was really nice to see. This was a cornfield, but every crop, of course, the soil's, pretty much the same in rotation. Sulfur was 7 ppm in this upper 6 inches and 9 ppm in this 6 to 12 inch acre furrow slice, which is borderline for having enough to grow most of our crops. Certainly potatoes, one of those. So we really thought we would see in some scenarios some sulfur benefits, especially knowing that potatoes basically don't have any roots below a foot deep. Now other nutrients in it, but other plants like corn, soybeans, wheat, they can mine these deeper nutrients from the subsoil where we have 31 and say 65 ppm sulfur. So we certainly expected potatoes to respond as well. But you probably know what I'm going to say considering I just said that. But so we put out a study just looking at some calcium sources, sulfur sources, really focusing on gypsum, calcium sulfate, which a lot of farmers are using already in their production system, to see what would happen. And we did this at different rates. Now we went a little bit higher. Depending on the producer for potatoes, they're using somewhere, 0 to say 1,000 pounds per acre. We have some questions because tomato growers are going upwards of 2,000 pounds per acre. Peanut farmers are upwards of 2,000 pounds per acre or so, depending on the producer. So we went a little higher here with the gypsum rates to see if it mattered. Overall, this is some work, from 2010 and 12. We did see, as tuber rots increased on this y-axis over the x-axis, rot decreasing with these higher gypsum concentrations, except in 2012. And coincidentally, 2012 was a high yield perfect year. So long story short, in good growing years, we really didn't see a benefit, at least in that one year. But if you had a more marginal high rot year, we did see some benefits. So, with sulfur rate, we never saw a benefit. I really thought we would. Largely, they were all flatline, no benefit to applying sulfur to potatoes. So that's one thing we're still curious about and working on. I see Austin's popped back on. I think I'm talking too much. So let's go on to the conclusion slide here. So overall, I know we talked about a lot of different nutrients and things here, but overall, we do need some phosphorus, even in very high soil test phosphorus concentrations, at least in our sandy loam, loamy sand soils in the mid-Atlantic region. We have some nice data to back that up. You can broadcast or band apply NP and K at planting from what we've seen in our studies and really don't impact quality. Gypsum, marginal years at least, where you have a lot of rot, it might improve marketability of some of your tubers. Good years didn't make a difference. And overall, we're still working on the sulfur thing. Crops are really hit or miss with sulfur, whether we see a response or not. And a lot of that's because there's a lot of sulfur still in that soil that we're mining and so forth. So with that, I want to thank the Virginia potato growers for sponsoring a lot of this work. Every year we have checkoff funds that they dedicate to us for researching different things, as well as the Virginia Department of Agriculture and Consumer Services, Virginia Specialty Crop Block Grants. Without those two entities, of course, this work would not have happened. So with that, here's our Facebook page if you want to just kind of follow us and learn a little more about what we do. And I post different things about extension meetings and something cool I see, I'll put on Facebook. And also, here's my contact information if you are interested in and have some more questions. So that's all I have, Austin, and I'll yield the floor to you.
Well, thank you, Dr. Ryder. Really, really appreciate you joining us today. A lot of good information there, and I'm sure that there will be some questions for you at the end here, so appreciate everything. With this, I think we're making the transition now to Dr. Yincho and Mark Clow. I think Grace and Aaron are working their magic on the Zoom side of things to make them the hosts. So, there we go. So I will turn it over to y'all. Thank you so much.
So Mark is going to give this talk for us.
Yeah.
And, I'll stay here for questions afterwards if there are any questions then.
All right, ready?
All right, Mark.
So this is a very, very brief update from Craig and I. And so thank you for the opportunity to talk today. The picture you see before you there is us planting the yield trial up at James Brothers just ahead of the COVID shutting everything down. There are 32 clones in that trial this year, 32 lines, and had I known COVID was on the horizon, I probably would have cut it in half, because the 6 people you see out there is probably about the same number we had to harvest it. So it was a daunting task. We got it done, and it was a decent year. So let's move on here. So this is the meat of the talk. There's a lot of content on this slide. So I guess what I want to start off in saying is there are 227 preliminary and advanced lines screened this year in our 17 yield trials. These yield trials were, the bulk of them, 12 of them, were located in Washington County here at the research station. 3 were in Terrell County, 1 in Pasquotank, which you saw the picture of, and then 1 in Pamlico. Within that, 3 of those trials at the research station were regional trials. 1 focusing on whites and yellows, another focusing on reds, 1 focusing on russets. 25 varieties total, so about a fifth of that 227 clones. There are 4 national trials we participated in. 1 was an early generation trial with the breeding programs across the country. Another was another national trial with not so early clones called the NCPT. That's actually 2 trials. It's Tier 1 and Tier 2. And then finally a snack trial, which is the advanced lines, most advanced lines. And a lot of times at this stage, those materials are going out to cooperators in large scale. So I put together this slide here with our the breakdown of the number of varieties we screened in the different classes, as well as some recommendations. NC 470-3 is our primary recommendation for a chipper. This is been a long time coming. It's going to be screened in the snack trials this year. So, as well as there's some bulk material going out to, I think it's Black Gold up in Maryland. So we're really excited about that. This is going to be a real good test year for it. And I'll talk more about that clone in a little bit. In terms of table whites, we out of 227, a fifth of those were table whites, and Brody was about the best clone that we saw this year for that class. Brody is later than Superior and Inval, but still a really nice clone, really nice variety at this point. Good set, good size. Set's a little bit low actually, but overall good yields because of the size. For reds, this last year I spoke about this clone. This year again, CO99076-6R. This guy is out of Colorado, and Real Potatoes has control of this one for marketing purposes. And actually, the picture in the lower right-hand corner is a picture of that coming off of our grading line and coming out of the washer onto the cull table. There's a little bit of skinning there, but this was a really nice looking clone. Yellows, Belmonte and Natasha are 2 I like. I finally got to see Colombo for the first time this year. I know some of you guys have been growing that for a few years. That was a really nice looking potato too, but I've got a lot of information and knowledge on Natasha and Belmondo. Belmondo's got a nice slick skin, very few defects, very few issues with it. Natasha is there because of the intensity of the yellow flesh. It's a lemon yellow. I mean, it's a gorgeous, gorgeous flesh color. I also want to point out the picture that's on the right side there, that's kind of dark. Those are our guys planting potatoes on the 19th of March. We planted till 9 p.m. that night, and that was the last day that we were allowed to plant. But we got everything in except for the beetle trials. So kudos to the research station crowd for doing that for us. The picture on the lower left is a picture of how we label bags for planting and some of the seed in one of the national trials. And the picture that I haven't talked about yet, that is the first steps in doing what's called genomic selection. And that is 1,028 lines or clone from our breeding program. That's all of them we selected in the first cycle of selection last year. And just a couple weeks ago, we took DNA from those, and first we're gonna get some trait data on them. You know, are they resistant to PVY and golden nematodes? But then a subset of those are going to be screened for this genomic selection process to hopefully speed up our ability to make crosses with better parents and select better progeny to get materials out the door. Right now, it takes about 12 to 15 years to release a potato. And so we'd like to advance that a little faster. So finally, I'll spend the rest of the last few minutes talking about 470. So this is a cross between Marcy and 182.5. And the really amazing thing about this clone is it's late vine. It is later than Atlantic. The vine is later than Snowden, but the potatoes underneath are really, they bulk ahead of senescence of the vine. So you get this strong vine holding up into the heat of North Carolina summer and protecting the potatoes underneath. It's reasonably resistant to IHN. We have seen a little bit in a year or two, I think, but it was like 1 out of 10 or 1 out of 40, and cut tubers. And a lot of times you're never sure because you're grading all white potatoes. One might have bounced in from another plot or something. And Austin says, "Shut up, Mark." So the other strengths are foliar late blight and moderate early blight resistance. It also is a pretty high yielding clone. You can see that in the bottom there, it says it's about 106% Atlantic. And its merit is, so the merit score is 1, 2, 3, 4 scale. 1 is outstanding, 2 is keep, 3 is marginal, and 4 is drop. This is 2.2, so 2 a keep. It's a really nice potato. It's going through the system, and hopefully we'll have some spuds available for distribution to a multitude of growers in the next few years. So with that, thank you for your time.
Wonderful. Thank you so much, Mark. Appreciate that update. And we'll keep it moving right along to try to stay close to our timeline here, so we have plenty of time for question and answer at the end. Dr. Gina Restaino.
Thank you. Let me go ahead and share my screen. Okay, so thank you for inviting me this morning. It would have been much more fun to be there in person to interact with everybody, but I'm happy to give you an update on some of the work we're doing on late blight, Phytophthora blight caused by Pinna kochianae, and some sensor technology that we're developing for detection of biotic and abiotic stresses in potatoes. So the overview will be, I'll talk a little bit about USA blight and then some of our sensor work for disease detection. Then I'll give you an update on Phytophthora kochianae work, and then we'll do the group questions at the end. So we know late blight has been a continual problem, chronic problem for many years in potato and tomato production in the U.S. The pathogen moves airborne through aerial spores or in planting material like potato tubers. And it has a polycyclic life cycle, so it can spread rapidly within a growing season, and if left untreated, can kill a field in a matter of days. We have a number of fungicides, good fungicides that are available for treatment of the disease. They're all listed in the Ag Chemical Manual for and the Vegetable Growers Guides for the Southeast U.S. We do have some fungicide resistance in some strains of Phytophthora infestans. So that's one reason why we genotype strains, to identify which ones might be resistant to the fungicide. And we know this pathogen can infect both tomato and potato. And in fact, many of the outbreaks in the eastern U.S. have been on tomatoes in recent years. And it can also survive in overwinter in wild hosts or in flowers like petunia. A group of us sequenced the genome a number of years ago, and we've used that genome sequence to do epidemiology work and population biology work to track the pathogen globally. And then, as most growers know, the varieties that are being deployed right now are susceptible to late blight. So that monoculture of susceptible varieties can exacerbate the disease, and that's why fungicide use is really important to control it. This is just some of the symptoms on potatoes, on potato tubers. We don't see the tuber blight as much in the eastern U.S., but there's some parts of the world, in Europe and in Mexico, where tuber blight is very common. And then the symptoms on tomato fruit and tomato stems. The pathogen produces these asexual spores of sporangia, and there are literally millions of them on a single lesion. So when those spores move from plant to plant, they can cause quick devastation in a field. Right now, we have one mating type that's dominant in the US, and we don't have sexual reproduction, mostly clonal reproduction by movement of sporangia. In 2009, we had a severe outbreak of late blight in the US that was caused by distribution of these plants from the Bonnie Company in tomato transplants. The disease quickly spread from tomato to potato, caused outbreaks, and really devastated the crops in the eastern US. The growers at the time that were growing these tomato transplants were not aware that they were infected, and due to that problem, this pathogen dispersed in the vegetable transplant trade. In recent years, we've been monitoring these plants more carefully, and they're being managed better, so we haven't had tomato transplant outbreaks. As a result of that, we developed a very large monitoring system with the USDA grant, which it's in its 9th year of operation now, USA Blight, where we monitor outbreaks, we map to the county level where the disease occurs, we have decision support tools, we're genotyping strains using microsatellite markers, and we're developing new diagnostic tools. So why do we need to understand what strains are occurring? Well, this just shows on potato just the shift in the different lineages that have occurred over time. Right now in 2019, 2020, we mostly have US 23, which is sensitive to mefenoxam, but these other lineages like US 8 and US 11 are resistant to mefenoxam. Most of the other lineages are also sensitive to the other classes of oomycete fungicides that we've screened. So knowing the lineage can help you determine what fungicide to spray. This just shows the outbreak maps from 2020. Many fewer reports of disease. Part of this, I think, was reflective of the fact that, you know, we're in the middle of not just a plant disease pandemic, but a human disease pandemic, and there are fewer people out in the field monitoring. We had fewer reports, and basically, you know, the situation doesn't look in terms of disease outbreaks as severe as in other years, but I think there's a combination of factors involved there. So I mentioned resistance. We have fungicide resistance in US 8 and 11. The other lineages are sensitive to mefenoxam, and they're also sensitive to these other compounds that we have tested. In Europe, they have found thiazofamid resistance in some lineages of Phytophthora infestans, and fluopicolide resistance has been reported in Phytophthora capsicum. So it's important to do baseline monitoring for some of these other compounds as well in the future. So what I wanted to spend some time talking about next is this newly funded project. We've been working on it for the last 2 years, myself and Dr. Qishan Wei, who's a member of my cluster, and he's an engineer from chemical and biomolecular engineering. And we've developed 2 classes of sensors that can be used to detect plant diseases. One is based on these microneedle patches. They're small polyvinyl patches about the size of a postage stamp with small microneedles. They're used to extract DNA from the plant, and then also we can do amplification and identify pathogen species from the microneedle DNA. And then we have this other second class of sensors, volatile organic compound sensors, VOC sensors. And I'm going to talk a little bit about each of those and how we're applying those for detection of not only late blight, but virus diseases, bacterial diseases, and fungal diseases on tomato as a model system. We were funded by the New Plant Science Initiative at NC State, and with that funding, we're really scaling up the sensor production. The microneedle patches are used to extract not only DNA, but we've been able to extract RNA. And that's a big breakthrough because RNA extraction from plants is a complicated process and can be expensive and difficult to do. Our microneedle patches is a small video, which I think you'll be able to see, but you press the patch on the leaf and essentially it removes the DNA from inside the leaf, the pathogen and the plant DNA, and then that DNA is eluted into a buffer, and then that buffer, we rinse the DNA or the RNA off that patch, and then we go directly to our molecular identification strategy. So it's very quick and easy, and we can scale up these patches. They're pretty much made in the lab with these molds, and we're changing the configuration of the patches. These patches, interestingly enough, have been used to immunize people. They're using them for vaccine, for like flu vaccine, and they've been used in the past for TB testing. The medical community has used these patches quite a bit. So there's applications for plant science as well. So the biggest breakthrough in the last year we've done, basically with Qishan and Rajesh Paul, our grad student, is to incorporate the patch technology onto a smartphone fluorescent reader. We've developed this glass slide, and essentially what we've done is develop real-time and a smartphone-based amplification of DNA from the microneedle patches on a heated glass slide that is then read through a smartphone. And we've done this not only for Phytophthora infestans, but we've been able to detect tomato spotted wilt virus, an RNA-based virus. It's a big problem, not only on tomatoes, but hundreds of other crops. It's a huge viral problem on many, many plants. And we've been able to successfully extract RNA using the patches and then using a qPCR method to amplify the viral DNA. And this just shows the progress curves on a real-time machine of virus amplification, P. infestans amplification, different concentrations. And then this is the amplification curves on the smartphone. We've done this work in collaboration with Anna Whitfield, a virologist who we've also hired in our cluster. Anna is shown here, and Qishan and his student Rajesh loaded the lamp primers up on this glass slide, so we can now kind of multiplex. This is a tomato gene, P. infestans, tomato spotted wilt, and a no template. And we inoculated healthy leaf, infected leaf, spotted wilt, infected leaf, late blight infected, and then combination. And you see how the color changes. This is healthy tomato gene, healthy and P. infestans, tomato in tomato, spotted wilt and tomato, and then both pathogens. So now we can simultaneously detect 2 infections by a virus and a plant oomycete in leaf tissue. We're scaling this up for alternaria and then for bacterial problems, because tomatoes get a lot of bacterial problems, bacterial spot and speck. So essentially we can make we can load the slide right now with 4x4, but we're going to go to an 8x8 grid so we can multiplex. So in 1 minute extraction, and then you run this on your phone and you get a readout. So it's a really amazing technology that we plan on scaling, and in years 2 and 3, we'd like to test this in grower hands. I know I've talked to some of the crop consultants in Eastern North Carolina and the county agents, and when we're ready to scale it, we'll be making some phone calls. So we've also developed a volatile organic compound sensor. This is shown here. using a gold nanoparticle paper strip that changes color based on what pathogen is present on the plant. And we've started this work out with Phytophthora infestans, early blight, alternaria, and then septoria blight. And essentially, over time, within a 2-day time frame, you can detect infestans before you see a leaf lesion. So it's really a powerful way of detecting pathogen infection before symptoms occur. And we're testing the volatile organic compound sensors now for the virus disease I just mentioned and bacterial diseases. So we want to multiplex the VOC sensors as well. And something that's kind of hot off the press, we're submitting the paper right now, but with Yang Zhu, who's in aerospace engineering and mechanical engineering, we're developing a wearable plant sensor. It's a graphene-based sensor that you attach to the plant while the plant is growing. And essentially, we can detect water stress and we can detect when Phytophthora infestan infects by changes in the resistance in this graphene sensor over time. So we're testing it on tomatoes right now. And the sensor it's called a Kurigami sensor. It's a foldable sensor, so it stretches and enlarges as the plant grows. And it will detect water stress. We're using it for detection of injury. So insect injury could be detected. And then infection by pathogens. And the sensor is kind of laced with nanoparticles that can detect many different volatile organic compounds. So this is really exciting as well, because this gives us the possibility of leaving the sensor on the plant, whereas the previous method I mentioned is paper-based, and it's a destructive sample. You remove the leaf and take the sample. And so all of this is kind of being integrated into a platform for monitoring emerging plant diseases. And the platform will include this database that reads the sensors, learns what the output looks like using machine learning algorithm for volatile profiles or for DNA based methods. And then we're going to incorporate this into a mapping system and a forecasting system for plant disease. We have the funding now from the Plant Science Initiative. We've also received funding from USDA and from the Keenan Foundation. So we're moving this work ahead, and it's pretty exciting. The other aspect I wanted to talk about in terms of important diseases that we see on potatoes in eastern North Carolina is Phytophthora nicotianae, and it looks very similar to late blight. It was described we first saw it in 2017, and the lesions look similar, but they're dry and they don't sporulate as much as Phytophthora infestans lesions. And we know that there is mefenoxam resistance in nicotianae, but in the dominant lineage of infestans doesn't have mefenoxam resistance. There's a soil-borne phase to nicotianae, no soil-borne phase to infestans, at least in eastern North Carolina. It's mostly in tubers and plants. And then we've got both mating types common for nicotianae and only one for P. infestans right now. And then we have symptoms on all parts of the plant, and then nicotianae can infect the roots, which is different. P. infestans does not infect roots. And nicotianae produces chlamydospores that survive from year to year, so potential for survival in soil is important. And it also has a wide host range. It goes to tomato, tobacco, ornamentals, very wide host range, whereas infestans is more specialized. So we found nicotianae in eastern U.S. and in eastern North Carolina for the last 4 years. We've been collecting as samples come into the clinic or through crop consultants or extension agents, and we are genotyping using microsatellite loci and mating type and doing fungicide assays. And pretty much, I haven't added the 2020 data on since we're still doing the mating type and mefenoxam sensitivity assays, but most everything's been the A2 mating type, except for in 2017, we saw both A1s and A2s. So in that year, we saw a mixture of mating types. But everything we've tested, which is good news, are sensitive to mefenoxam. And most of our samples are from potato, although it has popped up on tomatoes, so we're collecting from tomatoes as well. And then this just shows most of the mating types are A2s. And most of our samples are coming from North Carolina, although we've been collecting from New York and collaborators in Pennsylvania, and then also in Virginia on the Eastern Shore of Virginia. We've done genetic analysis using microsats, and these allow us to sort out multilocus genotypes. And this is sorted, the data is sorted by state. The red is North Carolina, and it looks like the North Carolina lineages are more diverse than some of the lineages we found in New York. These New York tomato lineages are shown here. And
We're getting a
then,
little short on time, Dr. Rustano. I apologize for cutting you off. I just didn't wanted to leave a little time for Anders.
okay, I have just like 2 slides, so I'm getting close. And then, so basically, well, I just wanted to point out that some of those tomato lineages are similar to the potato lineages, but then we have others. What we're seeing is geographic substructuring by location. You've got the North Carolina lineages, the ones from Pennsylvania, the ones from New York. So there's some genetic differences between lineages. And then this is a global population minimum spanning network, and the potato lineages all fall into this branch here, and our samples kind of fall into that branch here. These gray-green, greenish sage-green ones are our North Carolina isolates. So we're starting to understand where these fit into the global diversity and sort out mefenoxam sensitivity. And I just wanted to end by thanking many members of our cluster and our PSI team shown here, some of the folks I mentioned that are doing the sensor work and our virology work, and then my lab at NC State, and our funders here. And then I'll go ahead and leave it to Anders to end up the discussion.
Thank you, Dr. Rustano, for that update. Some exciting new technology coming that will really be great. And at this time, I'll turn it over to Anders.
Thanks, Austin. Can everybody see my screen?
Yes, sir.
Good to go? Okay. All right. Well, I'll be really quick. Today, I just wanted to cover a few things that are important for potato this season as far as thinking about some of the issues that we've got. So we're just going to cover a little bit about aphid management, also some wireworm control data that I'm getting out of sweet potato that I think is relevant for white potato as well. And so one of the new products that we're really focused in on, sorry, I'm having a little issue here. So one of the products that we've been looking at a lot in sweet potatoes has been Movento, which is an interesting product, oftentimes looked at as a nematicide. However, we do know that it has some activity on wireworms as a dual systemic material. So we've been looking at in sweet potatoes as far as how can we spray it on foliage and really reduce mid-season to late season root damage. We know that a lot of folks particularly in the Eastern Shore have had some success with this on white potatoes. Tom Kuhar at Virginia Tech has been looking at this product for quite some time. We think it has a really nice fit for trying to reduce some of the piercing sucking pest populations that folks have been experiencing. So particularly aphids, hoppers and mites as a foliar material, but also maybe some of this benefit as far as wireworm management during the season. And so I got some calls about aphids late in the season last year. So the one thing I wanted to cover as well is, how these materials might fit. We got about 2 years of efficacy data with this material in sweet potatoes. I think it translates really well. I paired it with neonics and diamides, so Admire, Platinum, and Verimark. Really what we see is, we can get adequate control of early season damage to either slips or mother tubers, especially when you incorporate an application of bifrenthrin as a pre-plant. So this is just a little bit of our results from 2020. What you're seeing is sweet potato data, but again, I think it translates. So the red arrows are really showing where we paired an upfront bifenthrin application with spinettaram or Movento. And what we see is better than average control relative to the untreated. We're beating or equivalent to chlorpyrifos plus bifenthrin. So I think this is a big win for the sweet potato world. But also that we do see really robust control when we're putting that foliar application down later in the season. So the material looked like a pre-plant incorporated bifenthrin application. So this would be analogous to what we would use for Platinum or Admire Pro and white potato. And then 2 shots of Movento 5 fluid ounces spaced 7 days apart. And what we see is some pretty good suppression of wireworm injury. The other thing I really wanted to cover is this aphid issue. So again, got some reports from various folks out east about reports of heavier than expected aphid pressure late in the season. I think that this is a real concern, especially for transmission of viruses around in the system. However, this late in the season, it's probably not necessarily that large of an issue. The question is whether or not it really confers any economic injury to the crop. And so just to remind everybody, here's our aphid threshold for potato. So early season, our tolerance for aphid infestations is 2 aphids per leaf. That goes up to 4 aphids per leaf after bloom initiation. And then within approximately 2 weeks of vine kill or harvest, it increases to 10 aphids per leaf. So it's really important to know where you are on that timeline as far as crop maturation and whether or not you need to make an application. The concern is when you have early season infestations, those populations can rapidly build and cause some real yield or quality issues for something like leaf roll. So there are several new products that are going to be out. So these are products that will be registered over the next couple of years. I thought it was important to really kind of highlight some newer things that are out there. So Savanto Prime is a material that's got good activity on aphids. Also has some activity on Colorado potato beetles. There's several nice formulations of this. However, a lot of these will need to be paired with surfactants, especially for aphids. We want to increase coverage around in the canopy and on the lower sides of leaves to really try to control these populations. Probably in a year, Safina will be registered. I've looked at it a lot in sorghum on aphid control. It seems to have a really nice activity profile for aphids. It kills a little bit more slowly than traditional materials. However, it seems to really get populations under control, soft and natural enemies, and it's good on a diverse set of aphids, including potato aphid. Obviously, there are several other materials that are already registered: Belief, Assail, dimethoate, Fulfill, Actara, and Amara Pro. The one concern here is that several of these materials for aphid control in potato are also neonicotinoids, and so we want to be mindful of whether or not we're putting additional pressure on potato beetle populations for resistance. We also know that green peach aphid have had resistance issues in the past. We don't really know where we're at right now as far as resistance in North Carolina, but it's something to be mindful of, especially with these neonics that tend to have some longstanding issues with resistance to a variety of different pests. And so just to be aware of the time, I wanted to stop and open the floor to questions to make sure you all have enough time to ask about fertility questions in potato. So with that, I'll stop sharing and remand the floor to Austin.
Well, thank you, Anders. Appreciate that presentation. And yeah, I'll open the floor up to questions here. I don't believe we've had any entered into the chat at this time, but for anybody in the audience, whether it was a topic that was specifically covered by the speakers, or if you just have a general fertility or potato breeding, disease management, insect question, I am sure that all the speakers will be happy to entertain those at those times. So open up the floor to any questions.
Austin, I'll ask one. I want to thank all the speakers for their great presentations. I have a question for Jean on the VOC sensors. How do those factor in? And I think I mentioned this to you before, but I know this is something you may have looked at since then. How does that factor in when growers are out there spraying fungicides? Does that affect the accuracy of that VOC sensor?
Yeah, good question, Erin. We are testing them now on tomato plants in the phytotron and in a growth chamber, but the sensors have to be able to withstand, especially the wearable sensors, have to withstand environmental conditions and also, fungicide applications, because tomato growers spray a number of applications of chemicals. One idea is to place the sensor on a trap plant or an indicator plant that's outside the field, because we don't want the sensor destroyed as a grower goes through, or, you know, tomato, they might prune or whatever. And in our second year of our field test, we're actually going to deploy them in Western North Carolina with Inga Meadows, in Waynesville and Fletcher, and see how they withstand the chemical applications. Obviously, we don't want to spray pesticides directly on the sensors themselves, because it will impact, I'm sure, how they read. So we're going to have to work through those kinds of issues as we deploy them in the field. And but you know, I can't answer it right now. I know if we cut the plant, or if we change the, you know, drought stress it, we'll get readouts in the sensors. And what we're trying to do is use machine learning to determine how these abiotic factors are impact the sensors. You know, fungicide application is something we could test in the phytotron, and we should do that probably with mefenoxam. I
Great.
can't answer it right now, but it's definitely on our radar screen.
Great. Thanks.
If you've got any specific questions that come from that, please reach out to your local county agent. You know, we're here to help you, so we can get in touch with speakers and facilitate answering those questions, even if it's not something directly from a speaker. Any questions at all you may have. But with that, if there's no further questions, I just want to thank all the speakers for taking their time to join us here today, and also thank everybody who got on the call. I know this Zoom and everything is new and weird for everybody, but we certainly appreciate you joining us here today, and even the people that are going to be watching this recording at a later date. Appreciate you interacting with us, and like I said, if any questions come from it, please contact the county agent.